Welcome back. In the last lecture I introduced cells. This lecture I want to talk about cancer cells. Last lecture, as you recall, I told you that cells are the basic structures of living things and that human cells have these internal structures. I introduced some of them. We'll see them now in the cancer context. And that human cells have all the genome, including cancer cells. This lecture I'm going to talk about the unusual properties that cancer cells have. And I want to emphasize that these properties, and I'll list them, and etc., are not present in all cancer cells, but many cancer cells have many of the properties. And I'll describe two of them in detail. One of them is that they divide without normal controls, and that's very important for the growth of tumors. And they don't die. That's something you may not have heard about before. So first, cancer cells' unusual properties. This is a photograph that I showed last time. It's a photograph of the cells that I study. It's a normal lung cell in brown in the background, and the kind of yellowish small cell lung carcinoma cell sitting on top of it. These are false color electron micrograph. Cancer cells can be studied in a laboratory in two ways. First, we can take biopsies from patients. Now, in the case of small cell lung cancer, the biopsy is relatively straightforward because people with this disease have cancer in their lungs, in the lining of the lungs, but the cells kind of are in balls and they lop off into fluid that'll be in the lungs. So a physician just goes in and takes some fluid out, and out comes these cancer cells. And here they are, the ball of cancer cells. And these are ones that were obtained, and I use them, study them in the laboratory from a 55-year-old woman with small cell lung cancer. Just to make sure that you're focusing in the right way, it's a ball of a couple of hundred cells. So here, for example, is one cell, and here is another cell. They're very small here, but there's a low magnification. But this is the way you can get cells from biopsies. A second way to do it would be to take cells that are normal in a laboratory environment, human cells, and transform them, add a cancer-causing agent. And what's going to happen when you do that is that the cells will become cancer cells. They'll overgrow, typically, and they'll look like cancer cells. In either case, we have to be cautious with any conclusions we come to. Generally, cells outside in a laboratory dish, outside of the organism, have more or less similar properties. But remember, the context of these tumor cells is very different. In the body, the tumor is surrounded by other tissues and fluids and hormones and all sorts of things. So the characteristics I describe from now on in this lecture of cancer cells, we have to take with a somewhat skeptical eye in some senses. Although when people have gone and looked at tumors in the body of an organism like a rat or something, a mouse, they find most of these properties there. The first property is that cancer cells lose all constraints to growth, growth meaning reproduction. In the lab, cancer cells overgrow. I just showed you that in the graphic that's currently there. In a tumor, cells detach in order to keep growing and to keep reproducing. They have to detach from this extracellular material that's around them. You remember? There was a protein made by the membrane that attaches cells to the glue that's outside of them. If there's a mutation and this protein gets changed, the protein no longer attaches. And that's exactly what happens as tumors develop. A second type of membrane protein I mentioned in the last lecture are proteins that bind one cell to another. If you change that program of proteins in binding, you no longer make a protein that'll bind two cells together. Now the individual cells are free to divide and move if they're metastatic, in other words, if they spread to other organs. Give you an example in clinical medicine. People studied a membrane protein called cadherin. It's one of those things that holds a tissue together in prostate cancer. And they related the cadherin content with survival. So here is a Kaplan-Meier curve showing, on the y-axis is typical Kaplan-Meier curve, showing survival, starting at 1 and going down, sadly, to year of diagnosis, after diagnosis. So we start off at the time of diagnosis at 0 and go on for several years. If the tumor had high expression of this protein that holds the cells together, you can see that the prognosis was much better than if it had low expression. So this property of cancer cells of sticking together is a very important one. A second property of cancer cells is that they have altered cytoskeleton. You remember the cytoskeleton, those fibers inside the cell. If you recall the cell I showed you a moment ago, remember the small cell lung cancers, round thing, and this flat pancake-like normal cell. If you take a photograph of us and stain for the in the light microscope for cancer cells and look at their cytoskeleton, it is really different looking. Here is a normal cell, and the cytoskeleton has nice fibers, and the cell has a certain structure. And here is a transformed tumor cell, and it looks like a mess. Alterations of these fibers inside the cell are important for the structure over shape of the cell, but also some of these alterations at the edge of the cell allow the cell to kind of have projection so it can move. This is very important in cancer metastasis, which I will describe in a subsequent lecture. That's the second property. Third property, cancer cells have altered cell communication. What do you mean? Cells communicate? Absolutely they do. There are cell membrane proteins that make a hole between cells. So here's the hole. Here's the actual picture of the cells. There's two cells adjacent, and there's a hole. They're little. You can see very small black holes here. And here's the hole here between two cell membranes. And here's the hole between two cells in a diagram. Now, why would a cell want holes between its in its membrane? The reason is it's a way of getting things rapidly from one cell to another, because it doesn't have to go through the membrane. You remember I described the cell membrane as a boundary to getting things through. It's inhibiting molecules. They got to pass through this fatty boundary. And so instead, we have these little holes, and things, small molecules, small substances like amino acids or ions, hormones, etc., can go very quickly. A good example is the heart. The heart has hundreds of millions of cardiac muscle cells. And you may note that the heart beats, all the cells beat at once. The ventricles beat at once. And they do that by signals spreading rapidly through these holes, through these pores. They're called gap junctions. In normal cells, typically the pores are intact. They integrate the cell very well, the signals. In tumors, very often, there are fewer pores between the cells. So the cells are not integrated, and they're kind of going their own way. This has great importance in therapy. If we have a drug that reaches the tumor on the outside, we want to make sure it'll go all the way to the inside rather quickly, and that's called a bystander effect of a drug. And if the cells don't have these pores, it's going to take a long time to get there. That's the third property of cancer cells. Fourth property of cancer cells is that they can spread to other organs. It's called metastasis. That's the subject of the next lectures. The fifth property, cancer cells can recruit their own blood supply when they're near the blood. That's called angiogenesis. That'll also be dealt with in a subsequent lecture. The sixth property I'll deal with a little more. That is, cancer cells lose or fail to differentiate. Remember, differentiation means specialization. Remember, stem cells, right, are undifferentiated in many tissues, and the stem cells become specialized. They divide, they form a group of stem cells, and when they're needed, because cells have been died or whatever, specialized cells have died, the stem cell stops dividing and becomes differentiated. Stem cells are pluripotent. They can give rise to a small number of cells. So the cells of the blood can give rise to blood cells, different types of blood cells, etc. They're in rapid cell division, so spontaneous or induced mutations can accumulate, not enough time for repair. So this can be a problem with cancer. But I'm talking now about differentiation. So here is a pluripotent stem cell, and then it becomes differentiated into various different types of cells. For example, here's a red blood cell and other differentiated and other cells. But in cancer, sometimes there's a blockage here. Some gene just doesn't get turned on properly, or a gene does turn on and stimulates cell division instead of differentiation. And so this cell that's on the way to being specialized says, stop, I'm not going to become a specialized white blood cell or red blood cell. I'm going to be a tumor. And this is exactly what happens in chronic myelogenous leukemia, which was the opening story of the entire course. Here is a blood stem cell, and there are two different lines it begins to differentiate into, called the myeloid and lymphoid cell. You don't need to know all these terms, except to know that on the way to white blood cells is a semi-differentiated cell called a myoblast, and that's the one that stops the differentiation pattern and instead divides, then divides and divides and divides and divides, and forms leukemia in the case of that patient, in the case of anyone with chronic myeloid leukemia. Now this relates, right, to the multi-step model. That's our seventh. This is the origin of cancer, is our seventh notion of cancer cells and what the properties are. And this is the idea that tumors are clonal. What's a clone? A clone is a group of identical organisms, right? Here come the clones. They're all the same. A clone of cells will be a group of identical cells. The assumption in cancer is, and has been, that all cancers come from a single cell. So here's the multi-step model, and we have a single cell being assaulted in some way, and initiation happens, and you get an initiated cell, and then more mutations happen, and you get a tumor, and it grows, and it metastasizes. All these steps in the multi-step model. Mutations pile up in a single cell. I've shown you a graphic similar to that many times in the course. If you do a survey of the cells of the intestinal epithelium in young people, you'll find a couple of mutations. Typically, if you're an older person, you'll find more mutations. And in colorectal cancer, you'll find 7 to 10 mutations. So they accumulate over the time of life. Now, what's the evidence for clonality? All cells of the tumor coming from a single cell? Well, the old evidence was you would look for a single mutation in different parts of the tumor. So you look in the tumor, you take a biopsy of this part of the tumor and that part of the tumor and this part of the tumor, and you look for a mutation in like P53. Remember that controller for cleaning up the genome. And you'd look for a mutation there, and you'd find the same mutation in all 3 places. Problem with that approach is you're only looking for single mutations. You're really not quite sure what's going on, but the assumption came about that said all cells of a ball of cells of the tumor are the same, so you got to treat them all the same way with a chemotherapy drug. But you know, people have known really intuitively that that's not so. If that was so, why do we need combination chemotherapy? There might be some cells with some mutations, other cells with different ones. The real question is, are all cells of the tumor cells as the tumor develops, or do some of them kind of lose the ability or never had the ability to be a tumor? One way to find this out is to do an experiment. And these experiments were done over the last decade or so, and they've come up with a very interesting result. You take a tumor, you break up the cells. It's like taking biopsies, only we're going to do it in an animal. So we take an animal tumor out, and we break up the cells. And we ask each cell, in an experimental setting, hey there, can you form a whole new tumor? So we'll implant the cell in a mouse or whatever and ask the question, can this individual cell, each individual cell, form a tumor? And the answer is, only a small number can. The other cells can't form a tumor anymore. What's their problem? They're not mutated enough, or they're dying, or we don't know, we're not quite sure. But the idea came forth that there are cancer stem cells, which are the important cells for forming malignant tumors. And that as a tumor grows, those cancer stem cells are the ones that are going to form the tumor, and the other cells just aren't able to form a tumor on their own. They're not as important. This has enormous implications for treatment. You see, if we just use chemotherapy or any kind of therapy, not surgery, but any kind of therapy on a tumor, and we say, let's kill as many cells as we can, we may not be doing it the right way. We may have to say, let's kill the stem cells, which are the important ones for growing a tumor, and the other ones will kind of die off over time. This is a controversial idea in cancer therapy. It's an idea that hasn't been taken up in the mainstream of cancer, but it's certainly an idea that researchers are very up on, and is a very active field. The heterogeneity of tumors, that cells are different, is now becoming very apparent with the sequencing of DNA in tumors as tumor genomes are sequenced. Because people are not just sequencing the genome of a tumor, take the whole tumor out and get its DNA. They're taking pieces of tumor, small fractions of tumor, because the techniques of DNA sequencing are such that you can do it on very small numbers of cells. And what you find is, if you look at different regions of the tumor, there are different mutations, multiple mutations in the tumor. But you know, in terms of multiple mutations, there's a model showing, you know, different colored beads, different colored cells. So the tumor itself ends up with very heterogeneous state. But you know, we've kind of known this all along in cancer treatment, because very often you kill 99.999% of the cells, and there'd be a small number left, and the remaining cells are resistant to chemotherapy. That's the case in the tumor that I work on, which is small cell lung carcinoma. You kill most of the cells, but the ones that are left are very different than the rest of them because they're resistant to the drugs. And by killing off the rest of them, you selected for survival only the cells that are resistant. In other instances, the resistant cells evolve, they develop over time. The problem in cancer treatment is therapy continues to assume that all tumor cells are the same. They don't have all these differences. So that's a real challenge as we go onward in cancer research and treatment. The eighth difference between cancer and normal cells. Cancer cells divide without normal controls. We'll talk about that later. The ninth, cancer cells don't die. We'll talk about that later. Look, it's time for a summary, because you probably lost track now of all these differences. So let me summarize the characteristics of cancer cells. First, they lose constraints to growth. Second, cancer cells have an altered cytoskeleton. Third, they have altered cell communication. Fourth, they can spread in the body, metastasis. Fifth, they can recruit their own blood supply. They have the ability to do angiogenesis. Sixth, they are not differentiated. They are undifferentiated. Seventh, they arise from a single cell, kind of. They're homogeneous, kind of, but really they're heterogeneous. They may be clonal, they may have come from a single cell, but tumors evolve over time. They change through time. Eighth, they divide without normal controls. And ninth, they don't die. Let me look at the last two in a little more detail. First, they divide without normal controls. Here's the cell reproduction cycle. Typical tissue. Okay, like skin, whatever, they're replaced by stem cells. And here's the cell reproduction cycle. It's under tight control. Cells divide only when they're needed to divide. Most cells in the body are differentiated. They don't divide. Remember the opening idea of control of the cell cycle in the previous lecture, where we had this symphony orchestra idea, right? And the upbeat, of the orchestra conductor, and the downbeat saying, let's get going, was this enzyme called a protein kinase, this protein called the protein kinase, discovered in yeast and frogs and present in other organisms. So we have the cell reproduction cycle here. It's in this circle like it was before. We start off with two cells, and each cell now goes to a point in the cell cycle where it's going to make DNA, which is kind of the point of no return. And then it divides here in the mitosis phase where you can see the chromosomes. Moving forward, we can look at the state of DNA during the cell cycle, chromosomes. Initially, in a cell cycle, when the cell is just doing its cell thing and not dividing, the cell sits there in what is called the G1 phase. It's just sitting there, nothing's going on, the DNA is not being duplicated. Here's after the point of no return, where the cell duplicates its DNA and it's going to make the chromosomes and then go into mitosis. There's a point of no return, which is called the restriction point. Remember, that's the point where both Hartwell and Mahler, these two discoverers of this kinase control, acts. It acts somehow to overcome a block or stimulate cell division at the restriction point. That's where it's going to act. What happens at the restriction point? So now we want to look at this in more detail. Now I'm going to give you an example of what happens at the restriction point, but bear in mind there are many different cell types and there are many different molecules that act at the restriction point. So the example I'm going to give is of one protein and one mechanism, but different cells have different mechanisms, and a lot of these have been worked out. The protein that acts at the restriction point is called RB protein. All right, so it's named after, not a person, it's named after a tumor. The tumor is called retinoblastoma. RB protein acts to block cells from passing the restriction point. The cell cannot pass the restriction point, and so it just sits there. And the cell going to sit there, it's going to be in this nuclear phase, GAP1 phase, and it's going to do cell things. It's not going to divide. So in most cells, differentiated cells, at most of the time, the RB protein is active, and the RB protein is blocking cells from passing through the restriction point, duplicating their DNA. Now the questions arise. First, how does the RB protein do its thing? How does it block cells from proceeding and making DNA? Second, how does the conductor, remember the downbeat conductor saying, okay, let's get going in the symphony. How does this kinase, remember the protein kinase, get around the RB block? Because that's where the protein kinase acts. So now we're going to look at the molecular biology of this, and I will show you very graphically the relevance of this to cancer. How does RB act? RB protein, it turns out, blocks the expression of genes involved with cell division and DNA duplication. The RB protein is a tumor suppressor. We'll talk more about this later on. I've mentioned tumor suppressors before. So what the RB protein does indirectly, it's not sitting in a cycle, is block the expression of genes. It grabs DNA, grabs onto DNA, and blocks DNA from expressing genes involved with cell division and DNA duplication. And as long as the RB mechanism is sticking on the DNA, then there's going to be problems, right? RB is a tumor suppressor. It stops cells from dividing. My second question was, how does the kinase stimulate cell division? Got to get around the RB block, right? So what does it do? The kinase essentially modifies RB. So it adds phosphate to RB. So here's RB indirectly stuck on DNA, and the kinase then modifies RB so it opens up and allows the genes for cell division and DNA replication to be expressed. It removes the RB block. They might ask, why are we learning all this molecular detail, RB and all this kind of stuff? Well, here's why, and this is, as I told you, I'm only giving you the example of RB, but there are many more examples we could discuss. Gene mutations in cancer. In some tumors, RB is mutated such that it doesn't go in the nucleus. It just sits out there, and it doesn't block the genes involved with the expression of genes involved with cell division. The result? Genes for cell division are expressed, and the cells go into the cell cycle repeatedly, repeatedly, repeatedly. No control. In some tumors, a substance made by the tumor binds to RB. So if RB is able to do this, but the substance binds to RB like a stick and prevents the RB from binding to DNA, its mechanism from binding to DNA. The result is the genes for the cell division are expressed and cells enter the cell cycle. So the RB mechanism, and there are many other similar mechanisms in nature and in tumor cells, is one that's very sensitive to things that can stop it from working. Some cells normally make blockers that inhibit the kinase. Remember, the kinase is going to take RB and it's going to modify it, right? Well, there's a protein called P21, protein 21. It's made in lung cancer. And in lung cancer, when you have P21, it blocks the kinase. And the kinase, remember, the kinase is the thing that opens up RB. But if RB can't get opened up because you're making P21, then there's a better prognosis. Well, here's an example, right? P21, it's a Kaplan-Meier curve of survival and years of diagnosis in a very serious type of lung cancer. And those lung cancers that make a lot of this P21, the RB sticks onto DNA because the kinase doesn't work, and so they have a much better prognosis. So far I've talked about internal controls of the cell cycle. There are also external controls. You remember a growth hormone or something can knock on the door and stimulate a cell division. So growth factors can do this by cell division. There are 3 stages to growth factor signaling. As you recall, the growth factor binds to the membrane receptor, activates it, it changes shape, and signals for binding are transmitted inside the cell. Many steps involved there, and then the cell responds. All of those steps get changed in cancers. We'll describe this in some detail later on. Cell division, for example, and the cell cycle and growth factors also has clinical relevance. Here are just some of the things. In some cancers, there's more expression for the receptor. So there's more receptors around the cell, and the result is, of course, more growth factor binding and stimulating cell division. In some cancers, cells make their own growth factor. So they just make it and make it and make it, and they self-stimulate for cell division. In some cancers, these are all mutations. There's internal signaling, all those steps inside the cell that go from growth factor knocking on the door to something happening in the cell nucleus. All those intermediate steps can be active and more active in some cancer cells. And so you don't need external stimuli. So there's a lot of relevance to this. Here's one example we'll come back to later on in the course. In some forms of breast cancer, there are more receptors. I mentioned that's one way for a growth factor. And there's a drug that's been designed called Herceptin that blocks those. And we'll talk about this in more detail later on. The ends of chromosomes turn out to be an interesting characteristic of the control of cell division. The ends of chromosomes are really interesting. Nobel Prizes have been given for the people who discovered this. At the ends of chromosomes is a special sequence called the telomere. Telo is the end. And it's about 50 to 200 times a sequence, a specific sequence, a small sequence of DNA. And it's kind of like it keeps the chromosome intact. Remember, chromosomes are huge molecules of DNA, 100 million base pairs of DNA. But at the end, there are about 16,000 caps of these telomeres, and they have proteins associated with them. I've given you an analogy. It's kind of like the ends of a shoelace, right? It's got this nice coating on the ends of a shoelace. Every time a cell divides and reproduces its DNA, this coating gets shorter and shorter. So imagine the plastic coating of a shoelace getting shorter and shorter, a little net loss of this plastic coating each time the shoelace is used, for example. And once you get to a certain point, it's so frayed after a certain number of cell divisions that the cell dies. The cell essentially is so unstable, the chromosomes are unstable, that the cell dies. So a typical cell in a laboratory dish will divide about 50 times, and then it'll stop because the telomeres get shorter and shorter and shorter. It turns out that cancer cells have a way, so you have these frayed shoelaces right at the very end, that cancer cells don't shorten their telomeres. How do they do that? They have a protein called telomerase it's called, and telomerase keeps cancer cells from dying. And it's an amazing mechanism because you need this in germline cells because you don't want chromosomes in germline cells to be shorter. So they have this telomerase that keeps the ends of the shoelaces intact. Stem cells, they're always dividing, so you don't want them to die. They don't. And cancer cells express telomerase. But normal cells, after a number of divisions, don't express telomerase, and they shorten their telomeres and they die. Well, we want cancer cells to die, don't we? So we'd like to block this telomerase. And in fact, there's a number of molecules under development for blocking telomerase. I worked on one of them. It's a component of green tea, which has been studied as an anti-cancer agent. The problem with the results of my own experiments were the dose you would need is quite high, but other people are working on it to try to get a dose that's reasonable. Blocking telomerase will block the chromosome from shortening and keep the cell stable. Cancer cells don't die. There is a process in nature called apoptosis, which is Greek for falling off, like of leaves. It's called programmed cell death, a series of events that kill cells, but it's a series of genetically programmed events inside the cell. The cell commits suicide. Why would a cell want to commit suicide? Well, if you look at the development of the human hand, for example, it starts off with webs between the fingers. And at a certain point in development, that tissue goes away. What do you mean go away? No, the cells commit suicide. In a number of instances, for example, cells in the body could accumulate mutations. For example, typical blood cells, white blood cells, get out into the circulation and they commit suicide after about 2 months. Why would this have evolved in humans? Why don't cells just last forever? The reason is they accumulate mutations over time, especially in the blood. A lot of things are in the blood. And in the linings of organs, the cells commit suicide. And it's a good mechanism because organisms that have that mechanism will survive better and be selected for reproduction because they don't accumulate cells with a lot of DNA damage. Now, normal cells that don't go into programmed cell death, that are just sitting there, have blockers of programmed cell death, obviously, because you don't want the cells to die. One of those blockers is called the BCL2 gene, and it's expressed in white blood cells. And when a white blood cell gets old, the BCL2 expression goes down. And the BCL expression goes down, of course, the cell will die. So it's altruistic. But in a tumor called follicular lymphoma, the BCL2 level is quite high. The cells don't undergo apoptosis, the cell stays alive. So the idea now is to target drugs that will inhibit the blocker, right? Inhibit BCL2, and some of those are under clinical trials. This is a really different way of looking at tumor treatment, isn't it? Normally we're thinking, let's stop cells from dividing. But, you know, a tumor growth means cells are growing and dividing, and cells are dying both, right? So we want to promote cell death, right? And a lot of chemotherapy turns out to be, and there are a lot of things I study in chemotherapy, turned out to be things that tip the balance towards cells dying. So if you damage a cell enough in its DNA, it will say, I've had it, I'm going to commit suicide. And that's certainly what we want in cancer cells. In this lecture, we've seen the properties of cancer cells, that they have unusual properties, that the cells, I focused on 2 of them, dividing without normal controls, and cells not dying. Now that we've got cancer cells initiated and we've seen what happens to cancer cell properties, the next 2 lectures will deal with the growth of tumors and their effect on people. Thank you.